In this video, we'll go over the answer key for Monday, April 13th. And our topic for today is transporting molecules across a membrane. And so we begin by looking at different type of ionophores. And ionophores are simply proteins that allow ions to go across the lipid bilayer. And you have two types: a carrier, which will grab your ion and physically carry it across the membrane, and a channel, which has a channel that allows ions to go from one side to another. Both of these speed up the ion by a lot because ions naturally cannot go through the lipid bilayer because they're charged, so they don't want to interact with the hydrophobic regions of the tail. Generally, each ionophore only carries one type of ion. Example here in vancomycin, which will only transport potassium. So our first question is just looking at our molecules here and asking ourselves, do we need a special ionophore for this molecule, which is mediated, or can it get through the channel by itself, non-mediated? So first, ethanol, even though it's polar, has an OH, it's actually non-mediated. The reason for that is it's small and it's just barely polar, so it can go through the membrane all by itself. While if we look at a molecule like glycine, which is relatively the same size as ethanol, now this is mediated. We need a special molecule to bring this across the membrane. And the reason is it's charged. Charged and polar are two, it's a big difference because charges really do not want to be without someone to be next to balance out their charge. So if glycine was in the lipid bilayer, these charges would have nothing to interact with, and that's very energetically unfavorable. So you need a transporter for glycine. Cholesterol, all hydrophobic for the most part, so you do not need a special transporter. So that's non-mediated. ATP, highly charged, big molecule, that you definitely need something to bring that across the membrane, so mediated. So let's look at another type of channel, which is multiporin. And porins are beta barrels. What a beta barrel is, it's beta sheets shaped in a barrel. And what multiporins do is that they help bring in maltodextrans, type of sugar. You have an hourglass shape here, which is a diameter of roughly 5 angstroms. And on one side, you have aromatic amino acids lining your channel. That's what's being shown here in yellow. So you have F, W, and Y all lining this channel. And what these aromatic side chains will do is that they will interact with the aromatic molecules of maltodextrin. You'll have base stacking. And this helps to bring the maltodextrins through the channel. You also have a bunch of hydrogen bonds interacting with the OHs of your dextran. So you have a perfect fit to bring these sugar molecules across. The aromatics are base stacking with your sugar, and you have hydrogen bonds to help them snake through this channel. The next type of channel we're going to look at is a potassium channel. And there's a lot of different potassium channels out there, but they are all very similar. And they're very selective in that they only bring potassium across. And the way that they do that is that they have a selectivity filter that will only allow potassium to go in. That is only something with a +1 charge in the same diameter. And the way that these selectivity factors work is that they have oxygens around, and only potassium can interact with the oxygens and move through. And also the pore helps as well, that when you get into the pore it goes through 3 angstroms, which only an ion the size of potassium can go through while interacting with those negative charges. So again, this is another example of a pore that has been specifically designed to only let one type of ion in. If any other charge atom ion like sodium or magnesium tries to go by, Either the diameter would be too different or the charges would be too different. Negative charge will not go near these oxygens. Something neutral charge will not interact with the oxygens as well. So we're making sure only potassium goes through this channel. So thinking about channels and thinking on how specific they are and what amino acids help them be specific, what amino acids do you think would be an abundant of a porin that is specific for a phosphate ion? So phosphate ions are negative, therefore only positive charge, or you would expect positively charged amino acids to be at the entrance. And for the potassium channel we just talked about, what type of non-covalent interactions are critical? Those would be your ionic bonds / salt bridges. So just as a reminder, these two terms actually kind of mean the same thing. And it is an interaction between a positive and a negative atom. And it's non-covalent. So these type of interactions are very important. All right, continuing on with the potassium channel, another question to make sure you understood this. Diameter of potassium channel can be 6 angstroms. Why can't water pass through it when it's much smaller? And that's what we just talked about. Channels are made for only one type of ion. So the potassium channel has these negative charges spaced out that will coordinate or interact with potassium channels only. Water is small, so it's not going to interact with these. Also, water does have a negative oxygen on it, so if you have a channel that's just like a circle of negative charges, water's not going to be able to go through that easily. So water is not going to want to be dehydrated, that is, lose all the other waters it can hydrogen bond with, to go through a channel where it can't really hydrogen bond with everything around it. It can hydrogen bond with some of the negative charges, sure, but the oxygen is going to be left out, and it's really too small to really get all the energy benefit of the whole pore. So how is a channel different from a transporter? So I got this infographic, so we know what a channel and what a transporter is. So a channel is kind of more like passive transport. It just opens up, allows ions to go through one side to another from high concentration to low concentration for the most part. And they don't use energy all that much. They don't really need ATP. While a transporter will use ATP, so they can move something across the concentration gradient, or they could move something across a charge gradient. And so that's going to be your active transport. So you're going to use ATP for a transporter. So that's the main difference when someone says a channel and a transporter. One uses ATP, one doesn't. All right, so let's look at how these channels are used inside your body, and we're going to look at neurons, which are just basically a bunch of voltage gated channels. What happens is when your neurons receive a neurotransmitter, that will cause a potassium channel to open. And when your potassium channel opens, potassium from the outside will flow inside the cell. This will make the outside membrane negative and the inside positive. And when that happens, your neighbor potassium channel will open and the same thing will happen. And what will also happen where the original sodium channel opened, a potassium channel will now open. So what will happen is that potassium which is inside the neuron, will now flow outside the neuron, restoring this positive negative balance. And we can look at this for a couple of graphs. So here we're looking at first the membrane. So on the inside of the cell is negative, right? So our potassium channel opens. So potassium permeability, our potassium channel opens. Our sodium channel opens. Our sodium channel opens, sodium goes in, and the inside of your cell at that area now becomes positive. We call this an action potential. When the sodium channel opens, the potassium channel now opens. And so potassium channels fly out of the cell. this causes hyperpolarization. That is, the inside the cell becomes more negative than it was at the start. So it's hyperpolarized, and it takes a little bit of time for that to return to normal levels. And you can see both the sodium and potassium permeabilities in the neuron. These channels aren't open for very long. This is in milliseconds, so it's very fast. But basically, this process of action potential and hyperpolarization goes along the whole cell till it reaches the end, and then we can shoot out more neurotransmitters and activate the next neuron or send some signal to some cell. So that's basically how a nerve works and what an action potential is. So how does the nervous system rely on voltage gated channel? Again, this question is just to check your own understanding. And as we said, we have a bunch of sodium potassium channels that run along the length of the neuron. These opening and closing helps to bring a charge across the neuron to relay signals. So let's look at the potassium channel in your neurons. So you can see that it's made out of 6 different helixes, and it's kind of weird looking in this picture. The helixes are really kind of in a ball or a cone, if you will, because remember, this thing's a channel. So we're showing a 3D image with a 2D representation, so they're really wrapped around each other like this. So the pore is going to be between channel 5 and 6, and what happens is that when you have the polarization, that is, we have our sodium channel open. Sodium goes from outside the cell to inside the cell. So the inside is now positive. On the 4th helix here, you have 4 arginine residues. These are positively charged. So what happens is that they move away from the charge on the inside, because that's positive now, and they move towards the outside membrane. When this 4th helix moves, your pore is going to be open, and so your potassium now flows out of the pore. This happens very quickly, and the pore is closed off very fast, though, because on the N terminus you have an inactivation ball, which are a bunch of amino acids that will go and seal up the pore. And eventually what will happen is that after all the potassium flows out, the inside becomes negative again. This 4th helix with the positive arginines will now be attracted to the inside, which is negative again, which will close the pore for good, and then you're back to where you started before the opening of the pore, before the action potential. And this slide just goes back and talks about this inactivation ball again, which I just said on the previous slide. So one other pore we're going to look at is called the aquaporin. Aquaporins move water through the membrane. Like every cell has an aquaporin. AQP1 is our best study aquaporin. And it's a big hourglass shape. You have a bunch of hydrophobic groups which allow waters to pass through this pore faster. And you also have arginines and histidines and backbone carbonyl groups that will make hydrogen bonds to this water as they move through it. You also have these asparagine residues in the center, which kind of works to like separate water from each other. And you can see this water is trapped between 2 asparagine, and it can't interact with water on either side of the asparagine. The reason for this is that water, when it's lined up, what it will actually do is that it will just start trading protons among each other. So if we start with one H3O plus, this hydrogen will go to this water. This water is now H3O plus. But then it will give the hydrogen to its next water, and so on and so forth. So instead of actually moving waters across, what will happen is that protons will just be moving across very fast. And you will have a buildup of protons on one side of the cell, which isn't good because protons are charged, and that can throw off a lot of your membrane potentials. So to prevent proton hopping, these asparagine residues help to block off water as it travels through the pore. So one of the things that we've been looking at for this section is structure and function. Structurally, let's look at these pores and relate it to how it can move these ions across. So that's one of the questions I would like people to know. How do structure and function of these channels work? So the potassium channel. Well, you have a bunch of positive amino acids, 4 arginines, and helix 4. When the membrane potential becomes positive inside the cell, these arginines will move to, not the new negative, but it's kind of negative, move away from the positive to the negative charge in the outside of the cell. This movement causes the pore to open, allowing potassium to flow out of the cell. Only briefly though, because the inactivation ball will come up and close the pore. So that's how the structure and function. The function of this channel is to open or release potassium during an action potential. The way it does that, the way it knows that there's an action potential happening is described here on our previous slides. Same with the aquaporin. The function of aquaporin is to move water across the membrane. So we have a bunch of hydrophobic amino acids that line the pore to help water move through it fast. We have asparagine residues in the center to help it prevent proton hopping. We also have positively charged amino acids to help, or just charged amino acids and backbones to help hydrogen bond the water as it moves through this pore. The last transport we're going to look at is GLUT1, which is a glucose transporter. So all cells need glucose for energy, so we need a specialized transporter to get those glucose across. So we're just going to look at GLUT1, and there's 4 different steps for this. 1, glucose will bind to the outside of the cell. When glucose binds to the outside of the cell, there's a transformational change. So originally the outside was open, but we have a change where now the outside, the pore is closed, and the pore is now open to the inside of the cell. Glucose will let go of the pore and go inside the cell. And when that happens, we need now to switch back. So the area that's open to the cell, that needs to be closed, and the area to the outside of the cell needs to be open now. So we need to have a conformational change. This mechanism, this opening and closing, makes sure that no small molecule could just slip on through, because glucose is a relatively big molecule in comparison to something like water or potassium. So if we just had a channel open to water or glucose only, you could think that maybe a water molecule, a potassium molecule could slip through accidentally. We don't want to do that. We want to control what is going in and outside the cell. So this mechanism of closing and opening allows us to control only glucose is going to go through, nothing's going to slip through. This is reversible though. You could accidentally transport glucose out of the cell. And in fact, there are cells in the liver that do want to actively transport glucose out of the cell. So that's just showing you how it could be done. And with that, that's the end of this video, and I will see you in the next video.